So liquid wastes contain high amount of ammonia, which can be really toxic for algae. And it's never optimal nutrient profile for the algal needs, so we need to basically rebalance this. And also, algae usually need some time for adaptation or grow on these particular nutrient sources. So in this, so I will show some results from our activities. So we are focusing on algae, what we can cultivate in big photobioreactor, up to 600 liters. And we test one marine strain, Chlorella minutissima, very good on bioremediation. Senedesmus, what is used a lot on mass cultivation in different locations, and photobioreactors and raceway ponds. And also we have some isolate from steel industrial site, basically a little bit similar approach what Sophie showed and talk us today. Then we isolate algae, what we think already adapt on some particular location, what is highly industrial site, for instance. And we use, like I mentioned previously, so agriculture waste, basically manure waste, trout farm waste, solid waste we use, rich in phosphorus, anaerobic digestion municipal waste, and we just like compare with F/2. So like you can see then initial concentration of nutrients, it's really high and we don't need to use that much. So basically we calculate how many milliliters by liter of culture we need to use, and it's really minor, so it just basically not that much what we need to use. So experimental conditions, so we grow algae with these nutrients in photobioreactors, and cultivation time was between 15 and 25 days. Some culture was, some experiments was done on batch mode of cultivation, some of them are semi-continuous to see how quickly waste can be remediated and replaced as well. So, and we did some close monitoring of biological parameters, water chemistry and biochemistry analysis in terms of provide data set for the modeling tool and fully understand the system with nutrients inputs and outputs and what we get during the process. And like we have these experiments and big cultivation system, we can take as much a sample as we want, and it's not limited by time, by volume and by time as well. What is usually happen when you do lab scale small cultivation experiments. So this is result of growth. We can see then Chlorella minutissima and Senedesmus growing quite well on waste and related to control. So basically it's around its control, and this gray points its waste. And also kind of on Senedesmus, this shows semi-continuous mode, so with replace of nutrients we can see culture can again take more of it and continue growth. So by nutrients uptake, it's also by measurements of total nitrogen and phosphate, we can see uptake is quite quickly happen on day even 5, so we can replace these nutrients and add some more as well to the system. And again, this is for the Senedesmus and Chlorella minutissima. So we also check for the synthetic efficiency, so what is, these parameters show kind of physiological response of culture, and it's reflect quite well as well than kind of needs of nutrients. And we can see in here then after 6 days, it's this for the synthetic parameters, it's dropped quite dramatically, and it's gain raised again when we add new waste nutrients. So focus on biochemistry composition, because it's really important to see not just kind of use this waste nutrients, but what is valorization of the biomass will be later on. And we compare quite a lot of trials together, and we can see then dairy waste, it's provide quite substantial, it's good biochemistry composition presented by protein, carbohydrates and lipids, mostly for protein and lipids in terms of Senedesmus, and Chlorella minutissima provide also protein and lipids high concentration. And these bottom graphs, we just basically compare these different treatments to see if it's any change during the cultivation process, or maybe we need to harvest earlier or later. And we didn't see any difference for the carbohydrates and proteins, but for the lipids, a substantial amount and profile is changing quite a lot during cultivation process, and related also on the nutrients what we are using. So this is results from fish waste, and again we can clearly see then algae adapt quite well on this particular waste nutrients, and they grow quite well, a little bit less intensive than control samples, but what is kind of normal. And uptake of ammonia and phosphate and phosphorus, basically uptake and build phosphorus in cells, was really positive. So this is kind of summary conclusion. We check nitrogen and phosphorus uptake during the growth. We calculate maximum productivity and production rate as well. And we can basically conclude an algae grows successfully on this particular waste. We have valuable biomass at the end, and it's kind of this result is comparable with control growth. This is what really good for us. Then we can replace these nutrients, expensive nutrients, by really free material. So another part of our work, like we have pack of photobioreactors, and we compare horizontal tubular system with vertical tubular system with different diameter, and we have some preliminary conclusion on what is kind of built on modeling and practical data as well. Then in controlling waste remediation condition productivity is higher in vertical system. This one. Maybe this configuration, we just would like to actually go a little bit deeper on the estimation to see design, and particularly light absorption and fluid and just adaptation on this particular system. And this is results was confirmed as well by some literature data, particularly in Wageningen University. They do exactly the same estimation, and they have exactly the same results. And if we are focusing on specific biomass composition, for instance, for example, rich on lipid, so this biomass can easily be obtained on the tubular horizontal reactor, and it's a bit more difficult to obtain on vertical system. So it's just kind of first approach. We are working on it to compare this together and get some results and usage of this particular different system in better way. So another part of this presentation, it's focused on photophysiology, and this work basically was kind of order, but to develop modeling tool, we need to focus on some physiological answer, and a lot of work was done on small-scale experiments, so it's flask. But when we start to grow with photobioreactor, situation is slightly different, and to double-check and practically see the difference, we focus on this research. So we use Nanocropsis oculata. It's marine strain, highly cultivated on all system for the mass production, and we use indoor photobioreactor 400 liters with different testing different photo period and to see how system answer on this different photo period. We use quite high intensity of light, what is mentioned here, and we didn't limit the system on any nutrients to basically just focus on photosynthesis answer. So this is a little bit of methods and materials. So we basically take samples daily for the cell count and pump fluorescent measurements to check the photosynthesis activities. And so this equipment we use, and we measure several photosynthetic parameters which provide kind of good indication for the cell health, trace, and light acclimation as well. And also the quantum efficiency of photosynthesis, very valuable measurements, and it gives these parameters to compare our results with other data set. And light saturation coefficient, so basically indicate how good this culture can adapt on this particular light, what is kind of even theoretical capacity of culture to absorb this light. So culture grow quite well on this slide, so a little bit lag phase of adaptation for this high light. And we did this on batch culture first. When culture reached 40 million cells per milliliter, we start to basically partially harvest reactor and replace the nutrients and see how these changes on nutrients can be reflect on photophysiological answer. So, yeah, this is lag phase. Culture completely recover from day 10 when we just and maintain the same pathway after harvesting. So our task was to repeat this 3 times to see before the acclimation. And this is another completely similar experiment that was repeated for the statistical analysis. And this is when we focus on photophysiological answer, we can see then FV of AFM, it's dropped dramatically after inoculation, and it's related to this adaptation of culture on the high lights, and usually it's answer like this. And when nutrients start to be limited as well, so physiological answer quickly show then culture need some changes and adaptation on these conditions, and it's improved after nutrients start to be in balance. And this is EK parameter, what like I previously mentioned, what is show capacity of culture to absorb light. And we can see then we provide this light at the beginning, and culture adapt quite well, and this basically pathway then culture can ready to adapt on 800 micromole photon per meter per second. So basically it's much more than we provided even. But it has some saturation in here, so it means then we can't increase this light anymore. And again, this repeat of the same experiments show a little slightly different results, and because of this basically photo period, like light-dark cycle was a little bit different. So by the conclusion, discussion and conclusion, so analysis of the biomass, it's in the early stage, and we will develop this analysis further. Our main target to check expression of protein D1, who's responsible for photosynthetic adaptation, and we will see on more like kind of molecular way how it was going, and it previously was not done. But right now, so we complete work on investigation of effect of photoperiod also to different, to using this different LED color mix. And we would like to combine some data of exactly measurement of the same parameters for the outdoor photobioreactor what we have in greenhouse, and just compare this. And the data obtained suggests then, despite the photo dose being the same, the increased light level results is a longer lag phase, and it's always kind of, you know, just time then culture need to adapt. And kinds of recommendation then lower light level can be used for the longer photo period, because we have this, our photo period was quite short, and high level light level didn't result in non-significantly higher carotenoid content, because we measure as well on pigments. So partial harvest has a negative effect on photophysiology, what is kind of we assume, but culture recover after nutrients have been supplied, and culture adapt to this new condition, and also kind of different harvests provide better, I mean, you know, just first harvest when culture just not used to it, the food acclimation quite long time taken, and after this is come too quickly. And so we adapt this for the kind of future reference of for the focusing on biochemical composition as well. So all this data go to the model, and also adaptation, and kind of better know our system, and all this data set will go for the modeling tool. So thank you very much for your attention, and this is my email of Naomi, so please contact us if you're interested.
you, Anna. Are there any questions for Anna?
Did you investigate, because you spoke about the composition of the biomass, you spoke about the lipid content. My first impression was that if you give effluent rich in nitrogen, you would have a decrease, or at least not an increase into lipids, because it's usually starvation that helps production of lipids. My second question is that most of the time we are also interested to know what happens to the metals, the heavy metals, and we know that algae can tolerate them and even absorb them. So are you investigating that also?
Yeah, very good question, thank you. First, yeah, nutrients, in particular nitrogen source in waste material, it's easily absorbed by algae, so it just gives the way to grow biomass, and after this it's become nutrient limitation, so it can boost to the lipid accumulation. So just we also did this kind of experiments, and I think so this makes sense. If we would like to have lipid-rich biomass, it can be easily obtained using waste from the nitrogen source. If I answer your question for this.
You put the effluent with the nitrogen and you stop.
Yeah, you can. Basically, we can program this. Yeah. If it's possible to do, because we closely monitoring nutrients concentration inside of the cultivation system and the biomass. So we just basically start to focus on it quite a lot. So we know which exactly stage, and when we should stop to produce the rich lipid biomass. Yeah. And second question for the heavy metal. Yeah. Yeah, it's one of our project, and what's related to an algae project as well. So we focus on measurements. We start to do it right now on heavy metals and even biological contaminants and the algal biomass after cultivation process with waste material to evaluate if we can use this for the feeding or for the others application. Yeah, but it's still in development. Yeah.
Still room for a question?
Yeah.
Somebody else?
How many harvests per week do you recommend?
Per week.
Yes.
When it's reached stationary phase, so basically every 3-4 days you can harvest. It depends also percentage of harvesting, so 25 to 40% you can easily do. Yeah. But you need to give time culture to recover what is